Let's talk about macrophages. Macrophages are part of the innate immune response. They are ready in a moment's notice to recognize a pathogen, attack a pathogen, and help the immune system. So let's go over some facts about macrophages. So macrophages are where are they found? Let's start with that. They're found in your tissues. There you can see I drew some green macrophages here. They're typically crawling around your connective tissues. You're not going to find them in your bloodstream. In the bloodstream, they're monocytes. Monocytes will enter tissues and differentiate into either macrophages or dendritic cells. We're talking about macrophages right now. So you're going to find macrophages living in your tissues. You're also going to find them living in your lymph nodes because, as we'll see, pathogens can get drained into your lymph nodes. So macrophages are phagocytes. They will phagocytose pathogens. And the fact that they stay in the same spot means that they're resident. That's the term we use. Macrophages are resident phagocytes. They will stay in the tissues where they're located. They're not going to travel. We'll see dendritic cells later are migratory phagocytes. Macrophages are resident phagocytes. What else do we want to talk about? So macrophages are going to do the three things we initially talked about when we introduced the immune system. They're going to recognize a pathogen, they're going to attack a pathogen, so they'll have an effector function, which is phagocytosis, and they're going to be involved in communication. They're going to release cytokines, which will help bring more immune cells to the site, which will help provoke a stronger immune response. First, I want to cover in this video how macrophages recognize a pathogen and phagocytose a pathogen. So here's a cut, a breach in the physical barrier, and here's some bacteria that enters the tissues. Bacteria can either be attacked by macrophages in the tissues, or the pathogen can be drained via the lymphatic system, via the lymph vessel, in the lymph fluid, and in the lymph node there are macrophages waiting to phagocytose that pathogen. So macrophages can work to phagocytose either in the tissue or in the lymph nodes. So now that we got that, let's talk about how macrophages recognize a pathogen. So we've got to talk about receptors. And just to review, receptors are proteins found on the surface of the cell, embedded in the plasma membrane, and they're typically going to engage with something in the extracellular fluid, in the interstitial fluid. So the receptors we're talking about in this video that are found on the surface of macrophages are phagocytic receptors. When they engage their ligand, that will trigger the receptor to undergo receptor-mediated endocytosis so that phagocytosis can occur. So here's a macrophage I've drawn, and there are some pathogens I've drawn on the right. So let's talk about some phagocytic receptors. There are a number of them mentioned in your book, but you don't have to know all of them. I just want you to know a couple. For example, the first one is the Mannose Receptor. So here's a pathogen on the upper right. In blue is a sugar called mannose. Mannose is a sugar that's commonly found on pathogens, not all pathogens, but some. And if the pathogen is covered in a mannose, recognized by the Mannose Receptor, which is a protein found on the surface of phagocytes such as macrophages, the Mannose Receptor will engage the mannose sugar found on the surface of pathogens, and that will trigger receptor-mediated endocytosis, and that pathogen is brought in via a vesicle into the inside of the macrophage. Once that happens, the phagocytic vesicle, or the phagosome, will fuse with another vesicle called the lysosome. The lysosome contains degradative enzymes, toxic substances, acidic substances, and when the lysosome and the phagosome fuse, they form the phagolysosome, and the pathogen will be destroyed. So that's the Mannose receptor. Mannose is a sugar found on human cells as well, but the Mannose receptor here doesn't engage the human mannose sugar. It engages pathogenic mannose sugar. Second phagocytic receptor is the LPS receptor. So LPS, you should have learned from microbiology, stands for a compound known as lipopolysaccharide. It is a compound found on the surface of gram-negative bacteria. So if you are infected with a gram-negative bacteria, your macrophages can recognize that bacteria. So there's some LPS covering that gram-negative bacteria. The LPS receptor will engage the LPS on the pathogen, and that will trigger receptor-mediated endocytosis, and that pathogen will be destroyed. Third class of phagocytic receptors are complement receptors, which we touched on previously. These are receptors that engage complement. So this goes to opsonization. If a pathogen is tagged with C3bs, complement fixation, then the C3b will engage the CR1 receptor, and that will trigger receptor-mediated endocytosis. So sure, if a pathogen is covered in Mannose and LPS, some phagocytosis will go on, but if they're covered in complement, you're going to have a much better efficiency of phagocytosis, and that is opsonization. There are other complement receptors, CR3 and CR4. They are going to engage with the complement protein I, C3b, which we haven't covered in the videos yet, but we're going to cover later. But I want to introduce that now. Something can happen to C3b that can be cleaved by proteins factor H and factor I to lead to IC3b, but that's going to be in a future video. But when IC3b is on the surface of a pathogen, it can be recognized by CR3 or CR4, and again, that's going to trigger receptor-mediated endocytosis and destruction of the pathogen. So we've covered in this video how a cell, such as a macrophage, recognizes a pathogen and helps eliminate the pathogen. We'll cover in a later video the cytokines released by macrophages.